Hi, everyone, and welcome to today's webinar, Automated Cryo-TEM Analysis of AAV Empty-Full Capsid Ratios, presented by Dr. Eric Bushong. We're excited to have you with us. We'll be reviewing the advantages of Cryo-TEM for characterizing AAV formulations and demonstrate how full empty capsids are reliably quantified with Cryo-TEM using our proprietary image analysis tools. Before we begin, let's go over a few quick housekeeping notes. The webinar will run for about 30 minutes, followed by a 15-minute Q&A session. You can submit your questions in the Q&A box in the toolbar, and Eric will answer them. If we don't get to your question in today's session, we'll follow up with you next week. A recording of the webinar will also be available next week. Now it's my pleasure to introduce today's speaker, Principal Scientist at NIS, Dr. Eric Bushong. Eric has a PhD in biomedical sciences from UC San Diego. He spent 15 years working as a project scientist at the National Center for Microscopy and Imaging Research at UCSD. He has extensive experience in transmission electron microscopy, scanning electron microscopy, micro-CT, and light microscopy. Please join me in welcoming Eric. Eric, over to you.
Hello, thank you for joining. The main focus of this talk today will be on AAV empty-full analysis using cryo-electron microscopy. Before going into that in detail, I'll begin with a brief introduction to how cryo-EM works and some of the general advantages to using cryo-EM for nanoparticle analysis. Of course, cryo-EM is probably most well known these days as a very powerful technique in structural biology, and Nanoimaging Services has extensive experience in determining protein structures. However, our company actually began as a characterization service, and we have almost 2 decades of experience in nanoparticle characterization. We currently operate 6 cryo-EM instruments across facilities in San Diego and Boston, and one of our facilities in Boston is GMP compliant. So what do we look at with transmission electron microscopes? TEM can be used to image a wide variety of samples, including proteins, viruses, liposomes, extracellular vesicles are seen here, lipid nanoparticles, virus-like particles, and inorganic nanoparticles. All the images seen here are examples of cryo-TEM, except for this image here on the upper left, and that's a protein seen in negative stain transmission electron microscopy. Negative staining is a more traditional approach to sample preparation for the transmission electron microscope. We choose between cryo-TEM and negative stain TEM depending on which approach best suits the sample and the analysis being. So the way that we prepare samples is shown here. For both negative stain and cryo-electron microscopy, one starts with a 3mm metallic grid that's covered with a thin support film. For negative stain TEM, the support film is continuous. The sample is deposited on this support film, and the particles adhere to the film. The grid is then coated with a thin layer of heavy metal solution that's allowed to dry out. And the heavy metal is deposited around the particles, and that's electron dense, while the organic nanoparticles themselves are relatively electron transparent, so you end up with images where the background is dark and the particles are bright. For cryo-electron microscopy, the support film is usually perforated with holes. It's sometimes continuous, but it usually has small 1 or 2 micron size holes in it. So after 3 microliters of the sample is placed on the grid, most of the sample is blotted off, leaving a thin layer of the sample trapped in these holes. And at that point, the grid is then very quickly plunged into liquid ethane, which instantly vitrifies the sample. And this vitrification process prevents damage to the particles, and they're frozen in a native hydrated state in this thin layer of ice. So in order to reproducibly perform this vitrification process, a robotic system handles most of the workflow. The operator places the sample on the grid, and then the robot performs the blotting and the freezing. And the parameters for the workflow can then be optimized for whatever sample is being prepared. In this slide, we can see example images of negative stain electron microscopy and cryo-electron microscopy, and begin to appreciate some of the advantages and disadvantages of each approach. With negative stain electron microscopy on top, you can achieve very high contrast because of the heavy metals surrounding the particles. And you can very clearly see even very tiny particles in these samples because of the high contrast. However, the resolution of the images is limited because the heavy metal stain itself limits the achievable resolution. In the upper right image, you can also see another limitation of the negative stain TEM approach, and that is the consequence of drying out the sample before imaging it. In this image, lipid nanoparticles that have been stained with negative stain versus in the image below it, lipid nanoparticles in Cryo-TEM. So the morphology of the LNPs in the negative stain preparation are clearly severely distorted and disrupted, whereas in the Cryo-TEM image, you see very nice preservation of the structure of each particle, and you can even see the internal structure of each particle and differences in the nanoparticle structure across the population. In the images on the left, you see AAV particles in negative stain on the top and Cryo-TEM on the bottom. So negative stain is a classic way of looking at AAV particles, and it's been used for a long time to determine the proportion of empty and full particles. But the interpretation of these images can be challenging because you're not actually looking at the inside of the particles. You're looking at how the stain interacted with the particles. So particles that are bright are interpreted as full because the DNA inside the capsid excluded the stain from entering the capsid, whereas the particles that are dark on the inside were empty and there was no DNA there to prevent the stain from getting inside the capsid, so they're dark. However, this can easily be a misinterpretation because if the capsids themselves are tight and well formed, they can prevent the stain from entering the capsid, and the particles that are empty will actually look full. It's also difficult to discriminate partial particles versus the empty and full particles using this approach. In the Cryo-TEM image of the AAVs below, you can see that the particles are now dark with a lighter background. And that's because any contrast in this image is coming from the particles themselves and not from a stain. So here the particles that are light on the inside are empty, and the particles that are dark are filled with DNA. And the interpretation of these images is much more straightforward, where DNA will make a particle darker, and the more DNA that's inside the particle, the darker it will become. It's possible to start discriminating partially filled particles based on the density of each particle. This slide generally shows the process of how we acquire data from a sample once it's prepared. You start by taking a very low mag map of the entire grid and then working your way up to higher and higher magnifications while selecting for areas that have well vitrified ice at the correct thickness. And once you've found those areas, then you collect the final high magnification images that are used for the analysis. The types of analysis that we can perform on these high magnification images varies depending on the sample type and the questions being asked about the sample. Some examples of the quality attributes we're often asked to assess are the morphology of the particles, obviously, and the loading status of particles, and assessing proportions of particles within a sample that have a particular morphology or are loaded versus unloaded. We can also perform particle size distribution analysis to show the range of particle sizes. And finally, we can look at other sample attributes such as particle aggregation, particle integrity, and the presence of impurities within a sample. So now that I've given an overview of how electron microscopy works and some of the advantages of using cryo-electron microscopy, I'm going to move on and start describing the method that we've developed for automatically determining empty-full ratios of AAV samples, and then also explain the steps we've taken to characterize the performance of the method. I'll begin by outlining the criteria that a sample needs to meet in order to be suitable for the analysis. The first consideration is the titer of the sample. We found that concentrations between 5E12 to 1E13 viral particles per milliliter are ideal. This range should allow us to directly prepare grids for imaging. If the sample has a higher concentration, it's usually straightforward to simply dilute the sample with a dilution buffer immediately before the vitrification of the grid. It's also possible, however, to image samples that are lower than the ideal concentration range, and we've been able to successfully analyze samples as low as E11 viral particles per milliliter. The grids that we use to prepare AAV samples for cryo transmission electron microscopy actually do have a thin continuous layer of carbon on top of them, and the AAVs have a high affinity for this carbon. So in general, we find that the presence of this carbon layer yields a nice even random distribution of AAV particles in any given field of view. But it also means that we can simply increase the absorption time that the sample is allowed to sit on the grid, and that increases the number of particles that adhere to the carbon layer. Additionally, we can also try multiple blots of a sample on a grid. And finally, if necessary, we can even just simply collect more images to eventually collect the number of particles that we're trying to collect for the analysis. A second important consideration is the buffer that the AAV particles are in. The most important aspect here is generally the presence of cryoprotectants. Sugars such as sucrose and trehalose reduce the contrast of cryo-EM images. Most formulations that we receive, AAV formulations, lack any cryoprotectants, and it's not an issue. But if we do receive a formulation that has some cryoprotectant, we can generally deal with that by simply diluting the sample with dilution buffer that lacks cryoprotectant immediately before vitrification to try and get the cryoprotectant below 5%, at which point we have enough contrast for the analysis. The next point is probably one of the strong points of cryo-electron microscopy compared to some of the orthogonal techniques for performing empty-full analysis, and that's that it requires very little sample to do the analysis. We can perform an empty-full analysis on a sample with as little as 25 microliters of sample. It only takes 3 microliters, as I mentioned before, to prepare a single grid. So with the 25 microliter aliquot, we have a sufficient sample to optimize the grid preparation and then prepare the grids for the actual analysis itself. And then as a final note, I'll say that we have experience imaging several serotypes of AV, including serotypes 5, 7, and 9. And we see no reason why the serotype itself would potentially affect the appearance of the particles in the cryo-EM images. And we've seen no influence of the serotype on the analysis results. So the first step in the analysis after collecting the images is particle picking to find each of the viral particles within the images. So you can see on the image on the left is a typical cryo-EM image with AAV particles. There are dark particles that are filled with the payload and light particles that lack payload. This is an ideal image with about 50 particles in the field of view. At this concentration, you're rarely going to see particles that are touching or overlapping. Occasionally it will happen though, and you'll want to avoid picking those particles. In addition, you'll see that here and there are small bits of ice contamination that are pretty much inevitable in cryo-electron microscopy. And so you want to avoid picking those ice particles or viral particles that are obscured by ice contamination. So we've developed an automated particle picking program that we feel does an excellent job of picking the AAV particles. Here we see the results of the particle picking process. We've assessed the accuracy of this picking process using the equation that you see on the box in the top of the slide. It considers true positives, false positives, and false negatives, but it ignores true negatives because that would overwhelm the accuracy score and make it meaningless. We've run this program on images that were collected of a sample that had mostly full particles and also a sample that had almost all empty particles, and it performs similarly well on both samples. We've also tested the robustness of the program for looking at images with different defocus levels. Increasing the defocus in the cryo-electron microscope increases the contrast of the image. So we found an optimal defocus that we like to work at that gives us a good amount of contrast for this analysis without negatively affecting the resolution of the images. But you can see that even if we purposely decrease the defocus by a significant amount, here from 6.5 microns to 4 microns, the program still performs very accurately. We've also assessed the sensitivity of the program. In other words, how many of the true particles is it detecting? And again, the performance is very similar regardless of whether you're looking at full particles or empty particles, and whether you have a higher defocus or lower defocus. So this is a program that's able to generate a high quality set of particles from an imaging session. It's not perfect though. We're still in the practice of manually curating the images after running them through this automated particle picking analysis. So that allows an analyst, an expert, to go through and quickly curate the images and remove any false positives or false negatives. And that gets us very close to 100% accuracy in the final set of particles that goes into the classification analysis. Nevertheless, to demonstrate the performance of the particle picking software, we ran the results of uncurated picks and curated picks through the particle classification process and compared the results. So on this table, that we looked at 4 samples ranging from one sample having mostly full particles to the 4th sample having mostly empty particles and a couple of intermediate samples. And in all 4 cases, if you compare the shaded row, which is the curated picks, with the white row beneath it, which is the uncurated picks for that session, that the classification analysis yields very similar results for all 4 samples. This seems to show that even with the few errors that are made with the automatic particle picking, given the high number of particles that we look at and the fact that many of the mistakes that are being made by the particle picking are false negatives, but as long as it's not biased or missing empty or full particles selectively, which doesn't seem to be the case, then that doesn't really affect the final results. So I move on to the actual particle classification process itself. where we're automatically assigning a classification of either dense, not dense, or partial to each particle. So I'll start by saying that we've taken a conservative approach to how we present this data in that we label the particles as not dense or partial. Because the fact is, we don't know for sure what the actual payload size is of any given particle. We'd love to be able to do that, and we're working on developing that ability. But for now, we know that within a given sample, a given set of images, there are particles of certain densities, and we can identify a population of the most dense particles and particles lacking any density and any partial particles in between those two populations. So in order to perform the classification, the images are first pre-processed to flat fill the background and remove any low frequency variations in the density of the background. The images are quite noisy, so we filter the images to reduce noise in the images. And then finally, we also normalize contrast across the images so that the intensity levels are consistent across the set of images. And then the coordinates that are result from the particle picking process I just described are then used to measure the density within each of the particles. And this results in many hundreds or thousands of density measurements. And at that point, an unsupervised learning algorithm then performs a one-dimensional cluster analysis on all these density measurements to find clusters within the data. And based on this, the clusters are classified as either dense, not dense, or partial. A final element of the analysis is the sample size. As of right now, we're in the habit of including a minimum of 1,500 particles for any analysis, but this would be a minimum, and there can be benefits to looking at a larger sample size, and I'll discuss that further in some upcoming slides. So that's how the classification process works, and I'll show you some steps we've taken to characterize the performance of the method. I'll start by looking at accuracy or specificity. We've been able to get our hands on a few reference samples, including these two reference samples of AAV5, one with mostly full particles and one with mostly empty particles. These reference samples were tested by the manufacturer using negative stain electron microscopy. So that's the baseline, the reference that we have to work with. When we run these samples through our analysis, we find that the results are very consistent with what would be expected for the samples. That clearly analysis has specificity for empty particles and full particles, and it can discriminate partial particles in between those two populations for both of these reference samples. Our results don't exactly match the specifications that were provided by the manufacturer, but in this case, I'm genuinely confident that our results are probably closer to the truth. Although the sample on the left, per the manufacturer, was 100% empty, we clearly see instances of dense particles in this sample. And likewise, the sample on the right was supposed to be 99% full per the manufacturer, but we saw numerous instances of empty particles in the cryo-electron microscope images. So this suggests that our results are probably closer to the truth, and it's not too surprising because negative stain microscopy can lead to incorrect results sometimes, and this is the reason why we prefer to use cryo-electron microscopy for AAVs. So the sample on the right here was used to test how many particles should be used in an analysis to get repeatable results. We had quite a few particles in this run, over 7,000 particles, and there's this small population of partials of about 3%, and we wanted to see how well we can detect that as we decrease the sample size. So we randomly selected sets of particles from this set of 7,000 some particles and tested how well the clustering analysis worked on these smaller and smaller sample sizes, and especially the ability to detect this small population of partial particles. And what we found is that the partials were detected down to at least 2,500 particles. And at that point, it became difficult to consistently detect the partials. But even down to as low a sample size as 500 particles, we were still able to consistently detect the empty and the full peaks. The dependence of the classification performance on the sample size is really context dependent. Here's another reference sample that we looked at that's now AAV9. And again, it was a sample that was supposed to be mostly empty particles. However, you can see in the images that there are occasionally a few dense particles in this sample. So we ran our analysis on the sample with only 1,200 particles, and we were still able to detect this small peak of dense particles within this sample. And so the take-home message is, if you're trying to detect a very small population of partials in between two large peaks of dense and not dense populations, it probably is going to require a larger sample size to consistently and accurately do that. But if you're trying to detect a small peak of partial particles, for example, in a population of mostly empty particles or vice versa, this is something that the classification analysis should be able to handle with a smaller sample size. So on this next slide, we're looking at repeatability and intermediate precision of the method. Here we have a single sample that we tested repeatedly on multiple grids. In the last 3 grids on this table, you can see that all the conditions were identical, same day, same lab operator, same type of grid. And for these 3 grids, we had an RSD of 0.8%. And if we included a wider range of conditions, a set of 6 grids with different days, different lab operators, different grid types even, we still had a relatively good RSD of 2.4%. So our intermediate precision and the repeatability of the method seems very good. So we were very fortunate to have obtained a series of reference samples that allowed us to carry out a linearity study. These samples ranged from approximately 75% full to 100% empty. All of these samples had been analyzed with multiple orthogonal techniques, including AUC. And here you see in the table on the left the results for the Cryo-TEM analysis, and you can see that the results show a nice linear analysis of these range of samples. And then on the right, you see the AUC results relative to the Cryo-TEM results, and it shows that there's a very strong degree of correlation between the 2 techniques. So this demonstrates how well the Cryo-TEM method can perform across a wide range of samples. And then finally, I'll discuss payload size. We've been able to look at samples with various payload sizes. Here you see the performance of the method with 3 different payloads. The separation between the dense and the not dense peaks is retained down to at least 2 kilobases. On the left, 5 kilobase sample, we had negative stain TEM as an orthogonal technique here, and I showed you before that the performance was very close to the expected values. For the sample that you see in the middle, the payload was approximately 4 kilobases, and we had AUC data as the orthogonal method, and there was very nice correlation between AUC and Cryo-TEM. And for the sample on the right, this was a 2 kilobase payload, and the orthogonal data that was provided by the group that provided the sample was mass spectrometry. And our results were very similar to the mass spectrometry results. Mass spectrometry found approximately 65% dense, 30% empty, and about 5% partials in between. The mass spectrometry and the Cryo-TEM results, if you look at the plots, they actually look very similar to each other, and the difference in the final results mostly appeared to be due to how the data was interpreted. So we feel that one advantage of our method is that the data interpretation phase is completely automated, and there's no room for subjective interpretation of these plots. The model goes in and it finds these peaks automatically. And in this case, there was no strong statistical justification for concluding that there was a distinct peak of partial particles in this data set. So that's what I have to present on the empty full analysis. I wanted to wrap up by just pointing out again that one of the strong points of cryo-electron microscopy is that in addition to performing the empty full analysis, there's also many other attributes of these samples that you can examine using the images that are collected in the same image session. So for example, you can detect instances of broken or malformed capsids in the sample. You can see free DNA that was either unencapsulated or appears to be escaping from capsids. And you can also see instances of aggregation and begin to characterize the nature and the size of aggregates that are in your sample. So it's possible to derive a wealth of information about your AAV sample from a single set of images. Thank you. And with that, I think we'll open it up to questions.
Thank you, Eric, for that great talk. Before we get into our Q&A session, I just wanted to draw people's attention to some content in the chat. There's our email address, so it's nischr@nanoimagingservices.com. Please feel free to reach out with any questions after today, or if you'd like to talk about any project in particular. A link to our white paper, our AAV white paper, is also in the chat, just learning more about our capabilities in that form. And for those of you that don't know, NIS is the only GMP compliant cryo-TEM lab in North America. And we'd be happy to expand on that and chat with you either today or after today's session about how we can support regulatory submissions. And along with AV, as Eric mentioned in his talk today, we perform cryo-TEM imaging along with analysis on a variety of nanoparticle formulations, including LNPs. And if you're interested in LNPs, we'd love to have you join our webinar on June 12th, titled Enhancing LNP Development, Cryo-TEM, and Automated Image Analysis Tools. And the sign-up link is now in the chat as well. So we'd like to move on to our live Q&A portion. And if you have a question, feel free to submit them in the Q&A box in the toolbar. And some of you have already done so. So the first question is about the ratio of empty full AAVs. Eric, so why is the ratio of empty full AAVs important? Is it a measure of DNA delivery capacity?
Sure. I'm by no means an expert in the clinical application of AAVs, but I think it's pretty well known that AAVs are not completely innocuous when you put them in a person, and there's always the potential for sometimes very severe reactions. And so it's considered a critical quality attribute to determine what proportion of the particles are actually carrying the payload in order to minimize as much as possible exposing the patient to capsids that are potentially going to have a toxic side effect without any potential for any kind of therapeutic effect. And so that's why people care about this empty full analysis. And yes, I think there's many different methods out there for trying to determine the empty full ratio of AAVs. And the reason that people have tried so many ways is because it's such an important thing to learn about AAVs.
Thanks, Eric. Our next question is about our software. Is our software GMP compliant? And then if you could speak to our turnaround time.
Yes, the software sort of environment that we work in at NIS is GMP compliant. We do GMP work for other clients already. We've been GMP for what, almost 2 years now, Sarah?
Yep.
I'll admit we haven't actually done GMP for AAV yet. We're still looking for our first client that we actually do validation of this for GMP. It would have to be on a case-by-case basis anyway, though, because every formulation that comes in is treated as its own formulation, and the method has to be validated for that formulation. So the way the process works is that we would go through a method development phase where we look at the formulation that's sent to us, and we establish things like the linearity of the method, the repeatability of the method, intermediate precision for your formulation. And in the process, the software is validated for being able to deliver the results that we expect. And then there's a final formulation phase where we collect enough data to show that the precision is satisfactory for the results that you need to have.
Just following up on, I guess, our analysis and the number of particles that we include. Is there a reason we use a certain number?
Well, I don't know if it's established yet. Again, that would maybe formulation dependent, case by case dependent on how many particles we need to look at to achieve the precision that you need for your analysis. I would say right now we always look at a minimum of 1,500. And generally that gives very good results, especially if what you mostly just care about is empty, full, and you're not worried so much about the partials. If you have a very small proportion of, let's say, 5% partials or less, and that's very important to you to characterize that from sample to sample, I imagine it may even be more important in more R&D phase work than actual final GMP study. But nevertheless, that might be a situation where we decide we need to have more particles in order to be able to confidently and precisely determine that.
I should say the next question. How are we dealing with overlapping particles or broken particles?
Our automated particle picker already is very good at eliminating most of those and not recognizing those particles, especially the overlapping particles, because there's a built-in logic to not pick particles that are too close to each other because we don't want overlapping particles. And then if there is the occasional broken particle, let's say that it's not quite complete, the capsid has a little gap in it, and maybe it gets picked by the automated picker. That's where I feel it's helpful to have the manual curation step where an analyst goes through and just quickly eliminates those few instances where that step happens. And just to eliminate, again, it's usually not many of those sorts of particles there, and they don't generally get picked. So I don't know if a few particles out of 1,500 is going to have a huge effect on an analysis, but that's why I like to do the manual curation to try and avoid those.
The next question. This is regarding where we were talking about the curated and traditional picks. They showed similar results. How did the curated picks differ?
Generally, we find more particles after going through the manual curation step because basically we're going through and picking some of the false negatives where a perfectly good particle, for whatever reason, sometimes does get missed by the automated particle picker. We pick up some of those and we add those. That's generally the biggest difference that I see between the manual curated step versus the initial automated particle picking is we actually end up with a few more particles after going through the manual curation step.
Are there other critical attributes that we watch for AAV, specifically regarding characterization under our GMP method validation services, aside from full empty?
For sure, again, we haven't done GMP yet, so I'm not sure what a GMP client might ask for that they consider a critical quality attribute and that they want to use Cryo-EM to assess for a regulatory agency. For our R&D studies that we do all the time for clients, we're very commonly asked to look at aggregation, to look for the evidence or presence of free DNA that's not being encapsulated. And for broken or malformed particles, dimers and things like that are very common in some of these formulations. So looking for the presence of those sorts of incorrectly misformed capsids. Those are probably the most common things that we look for.
Right. Have we used any other method, such as AUC, to verify how accurate our empty full percentages for
I,
our results?
I think maybe that was asked before I got to those slides. Definitely, yes. We've compared it to mass photometry and AUC and negative stain. And for all those three methods, at least, it seems to perform very closely to what the other orthogonal techniques report, especially AUC. I was very impressed with how close it was to AUC for that linearity study that I presented.
Regarding the negative staining, how is it best to understand that regarding the DNA that's inside? Could this prevent the staining buffer going where basically inside the capsid? Or could you speak more about the process of negative staining AV versus, I guess, in regards to cryo?
Well, that's the way that it's supposed to work for negative stain, is that the idea is that the DNA inside the capsid prevents the stain from getting in. So then what you actually end up with is a bright white capsid because there's no uranium inside of it. And so the bright white capsids are the full capsids. And then if there is no DNA in there, theoretically, the stain is supposed to be able to leak into the capsid and then turn the inside of the capsid dark. But sometimes that just doesn't work because the capsids are tight enough that the stain cannot get in. And so you end up with a bright capsid that looks like it's full, but it's actually empty. And so a lot of times, some of the times I'll say, the results that you get from the negative stain approach don't match what in cryo-EM.
For, again, the manual curation, I know we chatted about this before. Has our manual curation step enabled improvements for our automated process?
Again, the automated picker, it's not a deep learning tool. So it wasn't trained by humans for how to recognize the particles. It's more of a, maybe I'll say old school computer vision approach to doing it. So I don't know that the tool itself has learned from the curation process or that it could learn from the curation process. That's an approach that I'm curious about taking perhaps in the future. If we can develop a deep learning approach to doing that process, and then it might actually get better and better with these curated sessions. Right now, I think I've just mostly just learned that it's performing well enough, again, but about 98% accuracy, that the errors, they're not really common enough to, especially since there's no evidence of any real bias in the picking, empty versus full, that the automated curation seems to be doing a very good job. That's not the main thing I've learned from the manual curation, I think, really, honestly.
Good. All right, so switching gears to the images themselves, this is regarding someone asked about the density. So I believe this is from the images, but please ask again if this is not how the question was meant to be asked. But how is the density calibrated or normalized?
Yeah, the particle density. Yeah, I understand. So it's not calibrated. So basically, each report is its own data set, and we normalized all the densities within that study for those particles. I'm confident that the densities we see within a single imaging session are consistent because of that normalization process. We haven't yet figured out a way to calibrate the densities across several imaging sessions across weeks or months or years even, and be confident that the densities we're getting next year are going to be comparable to the densities we were getting on a session today, let's say. If we're able to do that eventually and convince ourselves that we're able to do it accurately, then we could potentially start to deliver maybe the payload and tell you, based on the density we see, we think that this is the payload that's actually in these capsids. We're not delivering that yet.
Thank you. Switching to more of the samples. So could we speak, or I should say, can you speak to the types of samples in terms of maybe upstream for the sample preparation process? So just the types of AAV samples that we can receive.
Most of the samples that we look at have been rather purified, more very downstream and in product. And so those are very easy to look at. I think that there's the potential to be able to look at more upstream samples that have more aggregation events, that have more maybe broken capsids or free DNA. I think this analysis would still work, especially because we do have the possibility of going in manually and curating and making sure that we have a good set of particles. Honestly, don't know for sure until people send us samples that are more upstream, how far upstream you can go before the buffer maybe becomes too high salt concentration to affect the contrast. It's something that we'd be very interested in trying with clients and to see how far upstream in the purification process we can look at samples and still achieve accurate results with the empty full analysis.
Great. Typically, how many grids do we look at per sample that we receive?
One grid generally is all it takes. We just make sure we look at enough particles from that one grid. We look at several areas across the grid to sample, but because the particle distribution across the grid is, as far as I can tell, completely random, as long as you're looking at enough particles, you should be sufficiently sampling the sample. And of course, we do screen grids to make sure that we have a high quality grid and pick a good grid for the actual analysis. And if we validate for GMP, of course, then the validation process involves looking at multiple grids and making sure that we have a consistent result across grids.
And just speaking to turnaround times again, I know we touched on the different steps, so the method development and method qualification validation. Could you provide just a rough estimate on how long that process would take?
Our typical turnaround is 4 weeks from the time that we receive the samples and all the paperwork is in place. We can definitely accommodate quicker turnaround times with rush processing. 10 days is something that we're typically asked to do, and we can potentially even do it faster if necessary. The actual analysis of once we are able to get the sample on the microscope, it's maybe a couple days to do the actual analysis and put the report together and deliver results. But it's a matter of getting the sample on the microscope.
Right. So the 4 weeks is more specifically to the routine testing, right? So routine R&D and routine GMP, whereas the actual validation process would be much longer.
Yeah, doing a GMP method validation would take much more than 4 weeks, yes.
Yeah. All right. I think that's all the questions that I see so far in our Q&A. I'll give it maybe one more second in case anyone has a last minute question. If you aren't able to ask today, please feel free to reach out to us. I know that our email is in the chat, but I would like to give a really big thank you to Eric. Oh, sorry. We have one last question. How long does a full analysis take?
I think I just answered that question. I would say, honestly, the actual analysis, I could do it all in a day. Yeah, sorry. Okay, yes. It doesn't take long to actually do it. It's a matter of being able to schedule the sample on a microscope and then do the analysis and put the report together. So the typical turnaround time is 4 weeks.
Yep. Wonderful. All right. And with that, I'd like to thank Eric again for this great presentation and for helping answer all of these great questions. Thank you to everyone who attended and for contributing. Again, if we weren't able to get to your question today, if you're watching this after, if you think of any questions to ask us later, please reach out on email. A copy of this recording will be sent out to all of those who registered. And with that, we'll say goodbye. Thank you again for attending, and we hope to see you at the next webinar.